Hello, in this short video I'm going to be talking about one of the protocols from the cell biology collection, the Apoptosis protocol. Let's start by opening the protocol. So from the Count Size tab, select the Protocols button. In the protocol browser that opens, protocols are arranged into collections. I'm interested in the cell biology collection and the Apoptosis protocol. I'm going to load the default protocol. The protocol loads on the left-hand side of the application. I'm also going to load the demo image set. This is easy to do. All I have to do is from the drop-down menu, click Load Demo. And as you can see, a 3-site multi-channel dataset opens. The Apoptosis protocol is going to try and detect an apoptosis marker within the nuclei of cells that are undergoing apoptosis. So we can see here that in well A1, there are quite a few cells that show this marker, whereas in A3, there are only a very small number. The first thing to do before executing any protocol is to click on the Sync button. This ensures that the protocol is aware of the channel names of your dataset. Now I'm going to choose the correct channel for nuclei. In this case, it's the Herxt channel. And then the correct channel for the target, which in this case is the Alexa 488 channel. I'm then going to click the interactive checkbox and click Next Step. In this step, Step 2, I'm going to attempt to successfully segment out my nuclei. Now by default, this opens at the threshold segmentation option. So I could just set regular threshold to try and find my nuclei. I could cancel this and change to smart segmentation. With smart segmentation, I have to select some example nuclei and then select a region of background. Image Pro is unable to segment out each of the nuclei in my image, and I can click OK. I can now configure the options for minimum area, clean borders, filling holes, and splitting. Let's start with the area. If I click Edit here, a dialog opens in which I can set thresholds for the minimum valid area and the maximum valid area. I'm going to raise the minimum valid area slightly. This excludes small bits of debris and noise from my analysis. I don't think I need to set a maximum valid area, as I don't have a problem with any large invalid objects. When I click OK, that change is applied. For clean borders, I'm going to choose the All option. This just means that any objects that touch the edge of the image will be considered invalid and not analyzed. If I click Apply, you can see now that I'm only analyzing whole nuclei. I'm going to set the Fill Holes option to On, and I'll leave the split at boundary shape because I think it's doing a good job of splitting my objects. I'll click Apply. I'm happy with this, so I'm going to hit Next Step. The goal of Step 3 is to separate labeled cells, or cells that are expressing the marker for apoptosis, with unlabeled cells. And by default, this is set to the Threshold tool. Again, I could click Cancel and change to Smart Segmentation if I wish. I think I'll be okay just to work with the Threshold tool to find those cells that are positive for the apoptosis marker and click OK. Again, I have a minimum area option here, which I can use to ensure that I'm only counting fully labeled cells and ignoring small bits of debris or noise. So here I can see I've successfully managed to identify my cells expressing the apoptosis marker. And I can click Next Step. I've now completed a single run of the protocol, and I'm in the Results step. Here I can see my results in the data collector table. I can see here that 8% of the cells in the field of view that I just analysed interactively are apoptotic. I can now configure other ways of displaying the results from the data collector. For example, if I look in the list, I can see that by default we're going to show the heat map and show the percentage of apoptotic cells in the heat map. That looks pretty good. I'm also going to show the block option and a graph. And I'm going to configure the graph to show on the y-axis the percent of apoptotic cells, and in the x-axis I'm going to show the collection number. In order to save these changes that I make to this list, I click the Read button. I could also choose to auto-create a report or auto-export my results directly to Excel. I'm now going to save my changes as version 1 of the Apoptosis protocol and click Finish. I'm now going to deselect the interactive step and click Run Selected, meaning we're going to run the protocol on all 3 wells. So let's do that. Here we see a progress bar, completed very quickly there. This is a multi-threaded analysis, so it's very quick. Here we see our heat map, in which we see that 81% of the cells from well A1 are apoptotic, whereas only 5 and 8% from the other 2 wells are apoptotic. We can see in the data collector table, we also see the percentage of apoptotic cells, the number of apoptotic cells, and total cells, and also a snapshot that shows us our analysed data. In my chart, we can see a plot of apoptosis. I'd also like to generate a report. From the report drop-down, I can select the default report template. So if we look in our protocols folder and look at the cell biology multiple sites, I want the apoptosis report. So let's click on that. If we click the report button, the report is generated. And here we can see the report that includes our heat map and a plot and our data collector table. If I click Save As, I can save this report as a PDF. And there's my report. And I'm going to click Finish. That's the end of my demonstration. If you have any questions, please contact Media Cybernetics, and we'll be happy to help.